next up on our agenda is Dr. Mark Halterman. Dr. Halterman is an MD PhD with a background in neurology and is the ADR for the Jacob School specifically, but at the same time, he is our go-to for everything downtown. So thank you, Mark, for taking the time to do this morning.
That's my new name, Downtown Halterman. I like it.
There we go. I like it. I like it.
Excellent. All right. Let me just pop the slides up. Jill, are you seeing the right deck?
Yep, it's perfect.
Okay, great. Thanks for the invite. So I have structured my comments on sort of a balance between not just what we have for core services, but what those cores will do. I assume the attendees are a range of investigator subtypes. Some may have no basic science experience, some may have all basic science experience, and our hope is that you might think about ways in which you could partner with clinicians if you don't now, or if you're a clinician, find a basic scientist that works in your area. So it's also been an interesting year, a lot of changes out of the NIH, and the institution is trying to be as nimble as possible and think about opportunities going forward. I appreciate Murthy bringing up the AI component. That obviously is going to be woven into a lot of the work that we are already doing and color what we do going forward. And it will, of course, help us sort of bend the arc a little bit with these cores, both on data analysis and the kinds of hardware that we're acquiring. As Jill mentioned, my background is in neurology. I'm a vascular neurologist, study stroke, and it's really a delight to be here at Buffalo, coming up on 2 years. So we have an interesting puzzle downtown at the BNMC, a lot of buildings. The cores are distributed throughout. We've got a lot of infrastructure already in place at the Jacob School at 955. The CTRC preceded us by about 12 years, not that long, 8 years. And a number of imaging modalities there and other resources, and I'm going to go through these in some more detail. The Center for Excellence in Bioinformatics and Life Sciences hosts genomics, spatial mass spectrometry, and ISO rooms. And then more recently, Hopman Woodward Medical Research Institute joined UB with a long list of really excellent resources. And I really won't have time to talk about them, but I invite you to take a look. I'm not sure if they're presenting to this format. Hopefully they are. So within the Jacob School, Pathology and Anatomical Sciences, a service center, as you would expect for a department of pathology, has things wrapped up in terms of tissue processing histology. We have confocal and flow cytometry, multispectral imaging, electron microscopy, and image mass cytometry. And let me tell you a little bit about each of those. So on the histology side, anything from frozen sections to getting your tissue paraffin embedded. We're developing expertise in creating tissue arrays. These are really exciting, and particularly for IMC, and I'll show you what that does for you in a few minutes. All of the staining you could ask for in situ hybridization and the equipment required to do so. We have made a concerted effort to improve infrastructure on the flow cytometry side, and we already had a very strong imaging side. So a lot of Leica equipment here, high-speed confocals, super resolution, and multi-photon for live in vivo work. On the flow cytometry side, several BD instruments, including an analytical flow cytometer, as well as flow sorting if you're looking to get single cell populations. More recently, within the past 12, 16 months, we purchased 2 Cytek Aurora instruments. These are spectral analyzers. Why is this interesting? It allows you to expand the panels of markers that you use in your studies. So instead of your typical 8 to 10 markers, now you're looking at panels of up to 35 different markers. And using the specific spectral patterns of these, you can really get a lot of data out of samples. So a lot more prep on the front end, but the data on the back end are extraordinary. And this just provides some examples of what that looks like. Similarly, we're moving from standard histology and immunohistochemistry into a realm of multiomics and looking at these slides in different ways. IMC allows you to take antibodies that have lanthides as tags on them and essentially perform mass spec on those flat sections. So your tissue is stained with those antibodies. A UV laser will ablate that tissue and it goes through a CyTOF mass cytometer and then reconstructs that image. And so the services in the core include conjugating antibodies that may be particular for your use case, although there are panels that are being developed commercially. And you can also perform flow cytometry on individual cells. So again, an increase in the number of markers you can look at, and then these data come together in really robust ways. So mapping the brain, for example, to look at 45 different targets is now totally within reach. And I mentioned these tissue arrays. You can take small punches out of a large number of samples and essentially satisfy statistical rigor in a very quick way. So many investigators are starting to take advantage of this, and we're delighted to see it grow. The core also has multispectral imaging. This takes into account label-free imaging across different spectra. This can be done at a microscopic as well as a macroscopic scale, and it allows you to look at simultaneous detection of multiple biomarkers for quantitative analysis. We have slide scanners in the core, several versions of this, and so the services include whole cell slide imaging, digital image annotation. There are databases to manage these images, and we're looking to grow the analytics side of image analysis, as this certainly is a huge footprint in the AI space. Electron microscopy is managed within the building. We have all of the equipment required to get your sample from initial fixation through imaging. So services, post-embedding labeling techniques, cryo-clem is demonstrated in the image below, allows you to look not just at the EM image, but also understand what's going on in other compartments within that cell. And this is a really important, obviously, modality for people that are looking at vascular substructure, they're looking at areas of tissue, and even organellar biology. So if we take it up a few levels, it's interesting to study tissues and cells, but we need to start putting together those messages into coherent arguments for how our interventions are working or what our disease targets mean. And one way to do this is through rodent behavior. This didn't exist at the Jacobs School. We received funding to be able to build several new cores, and rodent behavior is one of them. This is being led by Dr. Caroline Bass. So this will allow us to phenotype animals, provide training in behavioral analyses. The core will provide consultation services and will also conduct these studies. And so what I thought I would show you is just a couple of examples of what we have at our disposal here. So studies looking at, for example, mice that have genetic mutations in autism, and you can look at activities like marble burying. We have motor and cerebellar control assays like the rotorod, and then importantly, taking advantage of some of the genetic tools, specifically optogenetics, being able to flip a switch and create a behavioral outcome and study its importance in these neural pathways. You see the LED light has come on, and the mouse is now moving corresponding to the genetic network alterations that have been enforced. And then, of course, we are utilizing imaging and these kinds of genetic switches and optogenetic tricks to modify behavior in real time and use AI analytics, deep lab cut, and other software to be able to understand what exactly is happening on the behavioral side and analyze those data. So we are definitely in a new world with regards to animal modeling and what we can do. This just gives you a sense of the range of assays that are being set up in the core, covering learning and memory, anxiety, pain, and this is really a short list. So.
Thank you, Dr. Halterman.
Two more slides.
Oh, I'm
Neurophysiology.
sorry. Okay.
Neurophysiology is going to allow us to understand in real time what's going on in the brain of these mice. Rats also, you can look at turning on ensembles of neurons again, studying these in full depth in the cortex, and looking at how interventions are altering behavior and what that means from a network level. The NIH, if you've seen these announcements, is asking investigators to consider ways to not rely on animal models as strongly, looking at iPSC technology, looking at organoid models. So we are moving in that direction and reanimating cell phenotyping core. Currently, we have a system called the Cell Raft Air, which allows you to pick out clones based on imaging features, 3-color, and purify those and grow them accordingly. And we are moving towards developing microfluidics as well as a high throughput live cell imaging system that will take advantage of those, and then the software integration for image analysis and archiving. You're going to have the CTSI talking. I assume it's in your lineup, yes? Jill? Okay,
Yes,
great.
that's
Just to say that those of you that haven't visited the CTRC, you should do so. A lot of resources coming out of our Clinical and Translational Science Institute. I'm going to talk about a couple of them. First, the CBI, Center for Biomedical Imaging. We have both preclinical and clinical imaging opportunities. We have a Philips Ingenia 3T MRI scanner that was installed 2 years ago, state of the art. We also have a Brooker 9.4 Tesla for preclinical modeling that also has a cryogenic imaging coil, so you can do multimodal imaging, and you can see the services there. Again, where the field is moving is very much towards being able to bridge preclinical and clinical imaging and marry those with biomarkers. And so on that topic, we also have an incredible biorepository in the CTRC. Sample collection, sample processing, storage of said samples, and then we're moving towards federated models for distributing those and building research teams. So if this is something that could serve your work, please get in touch with us. We are very interested in growing our user base. I won't take you through the details, but you can see the equipment is there to satisfy those criteria. And then lastly, in the CBLS, we have our genomics and bioinformatics core. This is part and parcel with a good biorepository, but they do quite a lot of work in spatial omics. And then we also have mass spectrometry. The Center for Computational Research was mentioned, and there are several clean rooms. And that is the end of my presentation. Thanks so much for your time.
I'm so sorry to have interrupted you.
No, no
Apologies.
worries.
Apologies, but thank you, Mark. And as I mentioned at the start, when the senior associate deans and the ADR started to present, what we asked them to do was near impossible. I'm sure Mark could have probably delivered even more content over the course of a couple of hours, frankly, with all the resources that are out there. But he has a great team in the Office of Research in the Jacobs School. So I would also reach out to his team if you have any questions.